Can you hear me now? Please let me know through chat box. Am I audible? Yeah, thank you so much. We will go to this. Now I'm going to start from the first one. Okay. Okay, nothing but good morning, everyone. I'm welcome you all to this 17th day of CSIR Life Science Question and Answer discussion session. Today we are going to discuss about the part C questions of developmental biology. As per our schedule, the past 2 days and today we are discussing the part 5, the part C questions of the unit 5. Why? Because this unit carries more number of questions. Okay, this 4, 5 and 13 once repeatedly I'm telling. Unit 4, 5, 13 is very important for CSIR examination. If you are studying this three units thoroughly, means you can achieve 120 marks in CSIR examination. So please follow the commands, follow the questions. This is just that's refreshing session. In this session, yesterday and day before yesterday, we have discussed the sum of the part C question. Today also we are entering into the part C questions. Okay, now we will enter into the session. Cyclic AMP signaling plays a very important role in development and differentiation of Dictyostelium discordium. This is one of one organism. In this organism, this morphogen is synthesized by different adenyl cyclase enzyme. So as per the question, first line is a statement, just a hint, cyclic AMP role. And next one is the morphogen is synthesized by different adenyl cyclase expressed at a different stage of life cycles. Okay, this different morphogenes are synthesized by adenyl cyclase, which expressed at different stages of life cycle. So there are different kind of adenyl cyclase are present in that organism. The following statements PQRS, here they wrongly mentioned AD, here the PQRS referred to effect of mutation in different adenyl cyclase gene. Okay, what happened while we are mutate the this adenyl cyclase, means what happened in the cell. Okay, as per their here the correct answer for the A means the P and S statements are right. P statement, ACA deficient cells can allowed to aggregate by exposing them to the plus of CAMP. While you are mutating the cyclic adenases that make aggregation of this cyclic AMP and the ACA deficient cells. The second reason, the spores formed from the ACG deficient cells will germinate irrespective of the osmotic condition. There is a fluctuation, there is a modulation in this expression. This is the first question. So the reason, while you are mutating this adenyl cyclase enzyme that make a aggregation of the cyclic AMP and ACA deficient cell, and it's make a germination with irrespective of the osmotic condition. Next question, during earlier cleavage of cannot habits, elegans embryo. Each asymmetrical divisions produce one founder cell, which produce differentiated, descendant, and one stem cell. Okay, here this makes one stem cell and another differentiated in a descendant cells. Very first, very first cell division produce one anterior founder cell, namely AB. The one posterior stem cell, and namely P1. So posterior stem cell is called as the P1, and the anterior cells is produced by in the first cell division that are called as AB. When these blastomeres are experimentally separated and allowed to proceed further the development, one could get following possible outcome. What is the following outcome? They are given a research statement. In a laboratory conditions, they are doing this kind of researches, so they are included in this statement. As per their statement, what they are referred. Okay, in from the statement they are referred the P. They are doing a research with embryonic stem cells. This embryonic stem cells produce one founder cells which produce the differentiated descendant stem cell. So in the first cell division, they are producing one founder cell, one stem cell. The founder cell namely called as AB. The posterior stem cell is known as the P1. Oh, when they are separating the both. Okay, they are separated. The experimentally they separated the stem cell and founder cell and proceed further with the development. One could can following possible outcome. What is the following outcome? In this way, you can easily you can easily observe the command. Okay, P1 cells would develop automatically because that are the stem cells, while AB would show conditional development because there are founder cell. The founder characters are they need to maintain. Founder cells means that is the ancestor cell. So the founder cell, it should be that able to grow in only particular condition, that is the called as the, while AB would show the conditional development, and P1 cells are developed automatically, autonomously, sorry, it's autonomously, that is the individually it can grow, not depend anyone, don't need that are having specific characters. Stem cells are having the specific characters that if you want to develop particular organ, you can develop the organ from that particular stem cell. So that is that having this mutuality in any conditions. Next question: injection of Nakain's mRNA into one cell. UV irritated embryos of frog completely rescues cell development and allow the formation of complete embryo. Some of the following statements, that is PQRS, could possibly explain. So they provided this statement that the injection of mRNA into the one cell, UV irritated embryos of frog completely rescue cell development, allow the formation of complete embryo. So what is the correct statement? Okay, correct statement is P and Q, that is nocaine is secreted protein which induce the cell ectotherm. It's very important statement, they may ask in a 2 mark. Nocaine is a secreted protein which induce the cell ectotherm to form a neutral tissues, and it drosselize the mesoderm cells which would otherwise contribute to ventral mesoderms. Next term is nocaines binds directly to PMP4 and PMP2, thus preventing complex formation with their receptors. So this is the condition. It helps to it's allow. So due to this process, okay, PMP4 and PMP2 it's they are struggling, they are makers, they are not able to uh, bound with their receptor. So this is helps to dorsal development. It's allowed to form the complete embryo. There is no any attachments on there, so the embryo is fully grown. Next statement about the question in the next question, AP1, that is the APEPALA 1, it's a gene, is one of the floral meristem identifying gene. Very important thing here. Yesterday also we have discussed floral meristem identifying gene. In wild type Arabidopsis thaliana plant transferred with the AP1 and GUS. GUS is nothing but beta glucuronidase. This beta glucuronidase activity is since in floral meristem only after the commitment of flowering. After the commitment of flowering only this GUS shown activity. Ectopic expression of API and GUS is the embryonic flower mutant background result in a GUS activity throughout the shoot in a 4 days of old seeding. The 4 days, after the 4 days, they are observing this mutation. This observation suggested that AP1 is the stimulation of flowering in a EMF background. The D1 is the answer. So as per the question, once again I will read. AP1 is a gene which present in the floral this is the floral meristem identifying gene. Wild type Arabidopsis thaliana plant is transformed with API. They are transformed with API and GUS. That is activity is in this GUS playing activity in the floral meristem only after the commitment of flowering. Ectopic expression of API and GUS in the embryonic flower, that EMF mutant background result GUS activity throughout the shoot in the 4 day of old seeding. So they are observing the mutation GUS that they are observing this ectopic expression of API and GUS in the EMF mutant background. So what is the inference? What is the observation suggest that API is stimulation of flowering in the EMF background? Okay, next question. In case of CSIR gene, which of following is correct sequence of event taking place during the interaction of sperm and egg? Yesterday we discussed this story about this yesterday, a day before yesterday we had discussed. In case of CSIR genes, which of the following is correct sequence of event taking place during the interaction of sperm and egg? Okay, first one is the correct answer. A1 is the correct answer. Here, the as per the schedule, as per the life cycle of as per their circulation, they are having this unique characters. In that unique characters, the CR genes are having chemo attraction of sperm to the egg. What does the chemo attraction means? That egg surface having some receptors. That receptors are recognizing the receptors which present over the sperm head. Sperm head having some kind of receptors. That receptors are recognized by the uh, this egg surface receptors by soluble molecules secreted by the egg. Okay, so chemo attraction, that soluble molecules are attract the sperm cells and it helpful for the attachment. Then exocytosis of sperm acrosomal vesicle to release the enzyme that are releasing the enzymes with the help of exocytosis, binding of sperm to the extracellular matrix of the egg. After releasing of the egg, that signals are docked, that protein and the molecule released from the egg is bound. That bound combination passage of sperm through the extracellular matrix, and that penetrate into the that egg. Fusion of egg and sperm cell membrane happen. After this happen, that genetic material is transferred into the egg. These are all the sequential. First one is heme attraction, then exocytosis of sperm, then binding of sperm to extracellular membrane, and the enzyme release. Sperm passes through the extracellular matrix, and then eggs and sperm cell membrane. Okay, these are all the process which happen in the cells. And one more thing about this kind of question, we want to know the step by step process. Don't go with the this confusion mode. Okay, in this kind of question, just once you read the question and read the answer, just imagine the concepts. Then when wrist blastoma from recently cut axolotl forelimbs is placed on the host hind limb, cut at the end mid high level, it will generate only wrist. The host, those who own hind limbs was removed, will fill the gap and generate the limb up to the wrist. However, if the donor blastoma is treated with the retinoic acid on the grafting, the wrist blastoma is generated and complete lymph and will not allow the host to fill the gap. This happens because the retinoic acid. Okay, this is the paragraph information, this paragraph itself that having the answer. When a wrist blastoma from a recently cut axoloid forelimbs is placed on the host hind limb. Okay, this is the first statement. The mid high level. Okay, this is the mid height level. So that blastoma is placed over the host hind limb. Next statement. It generate the wrist, only wrist. It generate only wrist. The host cell. Okay, the host cell means that is the that host hind limb. This is the don't having the hist limb that was removed. That removed gap is filled with the lymph up to the wrist. Next statement, if the donor blastoma is treated with the retinoic acid on the grafting, the wrist blastoma will generate a complete lymph and will not allow the host to fill the gap. Why it's happen? This happens because of retinoic acid. It helps in the peroximalization of blastoma. Retinoic acid is helpful for the peroxidation of the blastoma and activates the HOX gene and differentially across the blastoma. This is the statement about this blastoma concepts. Okay, very easiest question, and just you want to logical question. In this question, they are provided the figure below represent a late zebrafish gastrula. The following concepts may be proposed during further development embryo. Okay, they are providing the stage and its differential stages. As per their statement, which is this true? As per the image, the small portion is the epidermis, neutral tissues, mesoderms, and yolk. This is the overall structure of the embryo. This structure, which they provided, the correct one. The correct one is P and S. The P is concentration of FGF decreases from the yolk towards the epidermis. Then, along with increases of PMP activity from the dorsal to ventral axis. This is the statement. Next statement, in comparison, both Xenopus and chick embryo requires activation. FGF for neural induction to occur in the addition to the PMP inhibition. Two reasons are the correct statements about this development of embryo. First one is concentration of FGF decreases from the yolk towards the excrement, along with the increases of PMP activity from the dorsal to ventral axis. Next one is in comparison. Both Xenopus and chick embryos requires activation FGF for neural induction to occur in the additional to PMP inhibition. Okay, next statement. When the prospective neuron from an earlier gastrula for a frog were transplanted into the prospective epidermis region, the donor cells differentiated into epidermis. However, when a similar experiment was done with the late gastrula of frog, that post prospective neuron development into neurons only. Observations could possibly be explained by the following phenomena. As per their statement, the first prospective neurons from the earlier gastrula of frog were transplanted into prospective epidermis regions. The donor cells differentiated into epidermis. So that is the concept here, that is converted into epidermis. However, when a similar experiment was done with the late gastrula, first step they are used the earlier gastrula, second step they used the late gastrula. This progress, that prospective neurons are developed only neurons alone. This observation could possibly be explained by the following phenomena. That phenomena are the P and R. Earlier gastrula show conditional development, whereas the late gastrula shows the autonomous development. The next option, the prospective neurons from the earlier gastrula, whereas they are from the late gastrulas, are determined. Okay, earlier stage are the very specified, but late stage it's determinants. Okay, next statement. In case of morpholytic regeneration, what is the morpholactic regeneration? That is the repatterning of existing tissues with little new growth that is happen in the morpholactic regeneration. Statement with respect to the extra embryonic structure formed in these mammals, as the functional attributes have been designated, that are allantoin stores urinary waste and helps mediated gas exchange. It is derived from the splanchnoplus at the caudal end of the primitive streak. The next statement is Q. Amanion is a water SAC, and it's protect the water sac that protect the embryo and surrounding the embryonic fluid. This epithelium is derived from the somatoplasias. Okay, somatoplasia. These are only two statements are correct one. The next question, decision of become either the trophoblast or inner cell mass blastostome is one of the first decision taken by the mammalian embryo. Belowing is a diagrammatic representation of different cells formed during the development from the morula with the help of different molecules. Identifying the molecule 1 to 4. What are all the 1 to 4 molecules sequentially? We want to get the correct one. So as per their statement, correct one is the COX2, CDX2. What is CDX2? This is the first one. This is the CDX2, which playing it's activating the epiplast and embryonic stem cell pathway. Then actor 4. This actor 4 is helpful for the morula to conversion of trophoblast. Then NANOG gene, which helpful to that embryonic epiplast and embryonic stem cell development and hyperplast. Then stat 3, it's totally helpful for the inner cell mass activations. Next question, the functionality of PAX6 gene is formation of optic and nasal structure may be attributed to following. The RNS is the correct question. Answers: PAX6 render the competent to receive signals from the optic vessels. The next one is, apart from the optic vessels, the head ectoderm may be induced by a PMP4 and CF8. So PAX6 is not exclusive for lens formation. So this this statement, this P and R are the statement for this question. As per the question is functionality of the PAX gene. They are asking focusing this PAX gene. So PAX gene is rendered the head ectoderm competent to receive the signals from the optic vessels, and apart from the optic vessels, the head ectoderm may be also induced by the PMP4 and GFG8. So PAX is not exclusive for the lens formation. The next question, an experiment in an experiment, sperm removed from the epidynamic of male mouse added in the dish containing the appropriate media and oocyte. No fertilization was seen, however, when a sperm from epididymis were directly placed in uterus of ovulated female, she become pregnant. So two statements are there. In experiment, sperms is removed from the epididymis of male mouse and added to the dish containing the appropriate media and oocytes. There was no fertilization happened, but they are remove the sperm from epididymis and they are placing that sperms into plant vegetative development. What does the plant vegetative development means? Q and S is the right one. Q is shoot oscillary meristem development this is the vegetative development. Shoot oscillary meristem development from shoot optical meristem during the differentiation of leaf primordia. Then another is a lateral meristem and cylindrical meristem found in the root and shoot results in the secondary growth. These two are the truth statements about the plant vegetative development. Okay, this is all about the unit 5. Today we are wind up the unit 5. I will send the notes for unit 5 also, and tomorrow onwards we will discuss the unit 13. Okay, so I welcome you all to the next day class, that is 18th day of our CSIR online question and answer discussion session. 18, day 19, day 20, we are going to discuss about the unit 13s. Okay, unit 30s or 13 also carries most number of questions from unit questions. Okay, part C we can expect 6 to 7 questions, so totally nearly 28 questions from this part C. So you can achieve the 7 questions from part C, so you can achieve the 28 marks. Another way, in that 2 marks are they are asking. So if they are asking 4 questions, if you answer the 4 questions, you can easily achieve the 8 marks. These are all the concept about the unit 4 and 5 and 13. So please study well. Tomorrow we will discuss the unit 13.